this is just a brief overview of the impact of genetic mutations on protein function. I go through some of the different types of mutations, and I thought it might help you with your lab. You're not required to watch this, but it's just an option if you guys are struggling a little bit. So in nature, mutations can be beneficial, but they also can be not so great. They can be harmful to an organism. For instance, this deer, he has an extra leg that could get caught, and it could prevent him from, running away from something. Or in something cool like this, which is the black panthers, that's actually a mutation in their skin coloring, and it causes them to be all black. So that's kind of a neat and cool mutation. In monarch butterflies, especially in Hawaii, which is an island, so this happens more frequently, is something called a monochrome monarch, which the butterfly ends up being white. And it has to do with the mutation of the color synthesis in the orange. Here we have a penguin that's white, albino penguin. It's through a mutation, and this penguin is more susceptible to like skin cancer and other problems because of his mutation. So what, let me move this little thing right here. So what do you think of when you hear the word mutation? So that's often the question I ask my students. And surprisingly, a lot of times people think of mutations, they think of Marvel characters, because the Marvel Corporation, the people that created Marvel and Universal, Disney, they did a really great job portraying the different types of mutations that occur in populations. So you have your mutations that you're born with, and then you have mutations that that can occur later on in life. So ones that you're born with, for instance, Cyclops here, he was born with his eyes like that. So he was already exposed. This was something he was born with. Spider-Man, on the other hand, he wasn't born with this. He was bitten, and so this mutation happened later on through his cells. Flash, he was struck by lightning, made him super fast. That was a mutation that occurred later on. The Hulk, that was an incident that happened in a laboratory, and it happened later on. And then you have Wolverine, which that would also be, he was born he was given that drug, so he would be a mutation that occurred later on. So how do biologists define mutations? So mutation is any change in the nucleotide sequence of DNA. So that nucleotide sequence, that GCAT, those different base pairs lined up. They may not always be lined up as GCAT. They could be GAT, whatever that base pairing lineup is. That is any change in that nucleotide sequence is a mutation. There's small scale mutations, which affect specific points along the DNA strand. And so we have a DNA strand. This would be a small scale mutation. It happens right at the specific points along the DNA strand. This would be a DNA molecule, and it would be a double helix because there's two strands winded together to form that helix. A large scale mutations affect large sections along the DNA strand. So the entire section is, affected by this mutation. There's different types of small scale mutations. There's substitution, where you have one base pair substituted for another. So you can see right here, this A was substituted for a G, right there. There's insertion, which we all of a sudden will insert an extra base pair or extra base pairs into a strand. And then there's deletion, when things get deleted, and then you end up with missing parts of your triplet kit or your triplet. These again would be small point scale mutations. Mutations caused by substitution. You can have silent, which has no effect on the encoded protein. You can have missense, which is the one that you see in the lab today, which is the change of one amino acid for another one. And then nonsense, which changes a codon for an amino acid into a stop codon. So there's something referred to, and you've probably learned this in lecture, called the central dogma theory. And the central dogma theory states that DNA goes from DNA to RNA to protein. So I like to refer to it as a recipe book, such as the one in this picture in this slide, where you have a recipe that has given directions. And then you wouldn't take, well, you shouldn't take your recipe book and take it from where you are and cook because you get it all dirty. You would most likely write down that recipe, and then you would bake it into something like a cake. So you have a recipe, it might call for 2 eggs, a cup of milk, 2 cups of flour, cup of sugar. And with all these ingredients, you wouldn't know. You could be making tons of different things, but you would know based on the recipe that you're baking a cake. So you would use that recipe and follow the directions to bake a cake. So you're translating that recipe into baking this cake. So there's some examples of some substitution mutations. So the protein known as brown chitin is responsible for making this coloring and of these this little moth. So this is the polypeptide chain that makes this brown moth. So you have methionine, proline, serine, valine, and you have a stop codon. So this is the amino acid, the mRNA sequence for this. You see it matches like that. But what if we accidentally changed out that G for a C? Like we changed it, right? So instead of having a C here, we changed it and made it a G. What happens? We have this instead now, right? We have CCC. We changed it. This would be a silent mutation because it still codes for proline. So if you go back, you'll see proline is up there, and you see that it still codes for proline. You have, and if I was to use my mouse here, you have C, C, and then C. That's how you read this chart. So you still have proline. So it didn't do anything. There's no harm, no foul. These do have, they can read in duplicate. So they can, like you can see here, several of them can read for the same thing. So all of these read for this one. All of these read for serine. So you can see they can read for multiple ones. They can have multiple reads. So this actually helps keep the function going if there is a little silent mutation. Still is intact, he has no problems. Let's say you have a min-sense mutation, like the one that was dictated, that we saw in lab, that we saw that that chain, that polypeptide chain you had, it swapped out the amino acid for it. So in min-sense mutation, you swap the substitution or whatever mutation that occurs, it actually swaps one amino acid for another. So here we swapped out the C right there. You see instead right here we had, this one was AUG, this was T, this one was going to form for our, this was going to form GGC, and this one actually we swapped it out for a C. So we're going to form AUG, GCG instead. So it's going to be GCG instead of what it should be, which is CGC. So what happens? We go here, and instead of coding for proline, now it codes for aniline. Let's see what happens. So now it's that instead of proline. It turned our moth pink. And this would not be the best thing for this moth, because the moth, so we could say aniline was actually, making it color brown, right? But if we now, proline was helping keep the moth brown, but now it's pink because it doesn't have proline in the amino acid chain anymore. Now it has aniline. It'd be like substituting flour for sugar. You're not going to have a good cake without flour. It's not going to work. Now you substituted out the proline for aniline, and now it's pink. This moth, this would be detrimental to this organism, because now instead of it being able to hide in the brown trees, now it's sticking out like a sore thumb because it's bright pink. Non-sense mutation is a premature stop while reading the translation. So right here we have our regular sequence up at the top, the AUG, CCG, AGU, and GUG. But what if we switch out in the third sequence, the A, we switch out this for this, like this. So instead of getting the UGA, instead of getting the AGU, we get UGA right here. We switched it out. We end up with a stop codon. So it doesn't read the full, it doesn't go methionine, proline. It doesn't read the full thing. It just stops prematurely. So we're going to go here, and it's going to stop. This ends up with a moth without any wings at all. So now it's a ground-dwelling moth. And again, this is not going to be beneficial to this organism. It's going to have issues because it can no longer fly. So what is the name of a mutation that changes an amino acid into a stop codon? This would be a nonsense mutation. What type of mutation is below? And you can see that I switched one out and it was a stop codon. This would be a nonsense mutation. These are just insertion and deletions. These are another type of mutations that can occur. It changes the reading frame. So if you insert it, we read it in triplicates, which is the codons. But if you insert one and you change the frame shift, it'd be like starting your sentence in a book on the second letter. Instead of reading like the, it would be he and then continuing on. So this changes the reading frame of the DNA sequence. I hope this helps. And if you have any questions, please contact me via email.